Hello everyone, in this video I'll be explaining on the topic fluorescent microscope. Coming to the principle of fluorescent microscope, most cellular components are colorless and cannot be clearly distinguished under a microscope. The basic premise of fluorescence microscopy is to stain the components with dyes. Fluorescent dyes, also known as fluorophores or fluorochromes, are molecules that absorb excitation light at a given wavelength, generally UV, and after a short delay emit light of a longer wavelength. The delay between absorption and emission is negligible, generally on the order of nanoseconds. The emission light can then be filtered from the excitation light to reveal the location of the fluorophores. This uses light of a high intensity source which excites a fluorescence agent, which in turn emits a low energy light of a longer wavelength that produces the image. The fluorescent light can be separated from the surrounding radiation using filters designed for that specific wavelength, allowing the viewer to see only that which shows fluorescence. Fluorescence microscopy uses a much higher intensity light to illuminate the sample. This light excites fluorescent species in the sample, which then emit light of a longer wavelength. The image produced is based on the second light source or the emission wavelength of the fluorescent species rather than from the light originally used to illuminate and excite the sample. The working of this fluorescent microscope is specimens are exposed to a light of shorter wavelength, that is UV light, which results in emission of longer wavelength, usually visible light. Light of the excitation wavelength is focused on the specimen through the objective lens. The fluorescence emitted by the specimen is focused on the detector by the objective. Since most of the excitation light is transmitted through the specimen, only reflected excitatory light reaches the objective together with the emitted light. Microorganisms in a specimen can be stained with a fluorescent dye. On exposure to excitation light, organisms are visually detected by the emission of fluorescent light by the dye with which they have been stained. Bacteria stained with fluorescent dye becomes visible as brightly glowing objects in a dark background. The parts or components of fluorescent microscopes includes fluorescent dye, or it can also be called fluorophore. A fluorophore is a fluorescent chemical compound that can re-emit light upon light excitation. Fluorophores typically contain several combined aromatic groups or plain or cyclic molecules with several bonds. Many fluorescent stains have been designed for a range of biological molecules. Some of these are small molecules that are intrinsically fluorescent and bind a biological molecule of interest. Most major examples of these are nucleic acid stains like DAPI and HOTJET, which is used to stain actin fibers in mammalian cells. The light source. Four main types of light sources are used, including xenon lamps or mercury vapor lamps with an excitation filter, lasers, and high power LEDs. Lasers are mostly used for complex fluorescence microscopes technique, while xenon lamps and mercury lamps and LEDs with a dichroic excitation filter are commonly used for wide field epifluorescence microscope. Next is the excitation filter. This excitation filter is typically a band pass filter that passes only the wavelength absorbed by the fluorophore, thus minimizing the excitation of other sources of fluorescence and blocking excitation light in the fluorescent emission band. Next is dichroic mirror. A dichroic mirror or thin film filter is a very accurate color filter used to selectively pass light of a smaller range of color while reflecting other colors. Next is emission filter. The emitter is typically a band pass filter that passes only the wavelength emitted by the fluorophore and blocks all undesired light outside this band, especially the excitation light. By blocking unwanted excitation energy, including the UV and infrared, or sample and system autofluorescence optically filter ensures the darkest background. There are two types, fluorochromy and immunofluorescence. Fluorochromy involves the non-specific staining of any bacterial cell with a fluorescent dye, while immunofluorescence uses antibodies labeled with fluorescent dye to specifically stain a particular bacterial species. Fluorescent microscopy has also been employed for detection of antigen and antibodies to identify structures in fixed and live biological samples. So this is about the fluorescent microscope.